they just... ¿Me lo apagas otra vez, por favor? Hi. So good morning, everybody. We are going to start this workshop, which is entitled Understanding Our Brain to Improve Therapeutics. Good morning to everybody, and I hope that you like this interesting workshop on brain functioning, and also how can we approach novel therapeutic strategies to treat neurological problems. Just to say that this symposium is funded by Fisher Scientific, and I would like also to thank the organizers of this conference, especially to Antonio Muro, who is, I think, is not here, and the rest of the organizers for inviting me to organize this workshop. I will be the first speaker talking on brain energy metabolism. Then Isabel Fariñas, we have the pleasure to have here Isabel Fariñas. She's professor in cell biology at the University of Valencia, and she's one of the best experts in neurostem cells in the world. So I think it is a high honor to have her here to talk about her work and recent work in this context. And finally, we have also the pleasure to have here Professor Giovanni Marsicano from the Neurocenter Magendie in Bordeaux, in France. And he's one of the best worldwide recognized scientists on endocannabinoid system in the brain. So we hope that you like this workshop. I will first speak on a topic, which is the topic that I am normally usually involved from the past 20 years approximately. We are trying to understand what are the molecular mechanisms that control the crosstalk between the different types of cells in the brain. As you know, the brain is a very complex tissue, and therefore, which is formed by different cell types, and obviously they are not working in synchrony. They are working in a cooperative manner. So there are factors, specific molecules, specific mechanisms that are controlling when a particular cell has to do a job and when another particular cell has to do another job in a collaborative manner. So trying to understand that is not an easy task because of the complexity of the brain itself. So normally the people use approaches, indirect approaches based on imaging, right? Normally the imaging, what gives you is a picture of changes in different areas of the brain that are connected to normally with the energy metabolism. And we shall see in a few minutes about this. Why it is important to understand this? I mean, the neurotransmission is a very highly costly process in terms of energy consumption. This is very well known since a long time. And why is that? It's because to reestablish the equilibrium of the ions across the membranes, in neurons and in astrocytes, there is an important need of energy, right? In the form of ATP. So ATP is consumed constantly in the brain, both in neurons and in astrocytes, in order to establish the ion equilibrium, all right? So this is something that has to be always in mind. And because of that, as I told you, the indicators of brain activity are actually based, most of them, on factors that has to do with the control of energy metabolism. For instance, the blood flow, the activity of the amount of blood that is going to areas that are activated is an indication of the activity of the brain. So this is meaning that supply of nutrients to a particular area is important. Also, specifically, you can also measure the glucose consumption. Glucose, as you know, is one of the most important substrates for most of the body, energy substrates, but for the brain, this is particularly important. So actually, you can measure by imaging the amount of glucose that is taken up by the cells, and that can give you several images. In red is more areas consuming glucose, right? In black or blue, areas consuming less glucose. And actually, it's not only that, but this is a picture that can be taken in different diseases, so you can diagnose problems in the brain, problems in neurotransmission, just by controlling, understanding what is the glucose consumption in the brain, right? But not only that, the third component which is important here is oxygen. Because oxygen, as you know, is the final electron acceptor of the mitochondrial respiratory chain, is highly consuming in the brain. This is very well related to the control of energy metabolism. You know, the brain is consuming about 20% of the oxygen that we breathe, of the air that we breathe. 20%. So it's a lot. I mean, the 20% of the oxygen that is in the air is consumed by the brain, and the rest to the body. But the brain is only weighs 2% of the body. So that means that the brain, when you compare with the rest of the body, is 10 times more active consuming oxygen. So all this picture is giving you an idea how important is the control of energy metabolism in the brain for controlling brain activity and neurotransmission. So understanding how is this control, because these pictures do not give you which is the contribution of different cell types in understanding this. So there have been many approaches to do that. So these, for instance, these factors can be also controlled in parallel, simultaneously. So there are several experiments. This is not done in my lab. This is done by other people, in which, for instance, you can stimulate in a very controlled manner, you know, the brain, the occipital brain, by just looking at particular image. And you can see that there is a perfect correlation between the blood flow that can be measured directly here. Can you see the red here, the blood flow going to this area? This is the occipital brain. And also the consumption of glucose here. This is in parallel. This occurs simultaneously. So it seems like they are connected factors, right? Blood flow goes there in order to provide glucose to the brain. However, very surprisingly, when you look at the oxygen consumption at the same time using similar methods, right? You can see that there is no signal at that particular time. And this is very tricky. This is not a failure of the system. This is not because you cannot detect oxygen, because when you wait for longer, you see that there is available oxygen here. So it's not a problem of the system not detecting oxygen. It's just that simultaneously with the blood flow and the glucose consumption, oxygen is not consumed. However, we know that oxygen is consumed to the brain, by the brain, and very importantly, and is essential for life and for the brain function. So what's going on here? So the interpretation, the best interpretation of this is that, paradoxically, because we know that we consume oxygen, glucose is being consumed through the anaerobic pathway by anaerobic glycolysis. You remember that anaerobic glycolysis is a transformation of glucose to lactate without passing pyruvate to the mitochondria. But this is a paradox, because we know that besides glucose, we are consuming oxygen, right? However, it doesn't take at the same time. So it is not that the case. Oxygen, indeed, is being consumed by the mitochondria, right? It is being consumed, but there are different cells which are contributing to both things. There are cells that are specialized in consuming glucose, and there are other cells that are specialized in consuming oxygen. So this picture, the aerobic glycolysis, is actually, I don't know why it goes so fast. Yeah. Is actually separated, compartmentalized in the brain, right? There is a compartmentalization of the brain metabolism. And the experiments leading to this interpretation have not been easy. People have to take advances of cultures, of very fine approaches in vivo, and many other techniques. For instance, you have to go to molecular mechanisms as well to know what are the molecular mechanisms controlling this factor. For instance, in the astrocytes, it is well known that they are highly glycolytic, anaerobic glycolytic, right? Anaerobic glycolysis means that glucose is being converted into lactate regardless whether there is oxygen or not, because it is not being consumed by the mitochondria. And we observed our own contribution to this field was to identify that actually there is one particular protein which is an activator of one of the key points of the glycolytic pathway, which is a PFK-FB3, it's a phosphofructokinase fructose bisphosphatase, which makes an activator of this pathway. And the expression of this protein is very high under normal conditions in the astrocytes. This is a Western blot showing the high expression of PFK-FB3 here. Can you see? Right? However, if you look at neurons and the control conditions, look at this pathway, it's virtually absent. Right? However, if you treat the neurons with a system that stabilizes this protein, okay, here, and you measure the rate of glycolysis, right? The rate of glycolysis using a radioactive method that converts glucose into tritiated water, you see that neurons that were usually very little glycolytic, they start to perform a lot of glycolysis here. Can you see? Just simply because you are stabilizing that protein. So the conclusion of that is that neurons are completely different from astrocytes in the way that they conserve energy. In the astrocytes, they prefer to use glucose glycolytically to lactate to obtain ATP in the cytosol, and that appears to be sufficient for their survival, whereas in neurons, they prefer to use pyruvate in the mitochondria, consume oxygen, and produce ATP by a different approach. So here we are. What are, therefore, neurons doing with glucose? Because besides astrocytes, glucose is also used by the neurons. So the neurons, instead of using that glucose in order to make energy, what they are doing is to convert into NADPH and reduce glutathione, which are antioxidants. And that is something that happens through a different alternative pathway, which is the pentose phosphate pathway. So neurons do prefer to consume glucose through this pathway instead from the glycolytic pathway. So therefore, what is the source of energy for neurons? We know, we will go to that this is even more interesting under the therapeutic point of view. When these neurons, you treat them with an insult, which is a situation which is common to different neurological diseases, there is a pathway that stabilizes that protein, which is the proglycolytic one. You can activate glycolysis in the neurons, and you actually can promote glycolysis in the neurons artificially in the lab, in the neurons, in the cell culture. However, when you do that, there is a compromise in the use of glucose through the pentose phosphate pathway because there is not enough. There is a competition between this pathway and glycolysis for glucose. So when you stimulate glycolysis, there is a reduction in the use of glucose through the pentose phosphate pathway. And that is therefore causing an impaired ability of the cells to reduce glutathione, and therefore there is redox stress and there is neuronal death. And that is a mechanism that we propose for some neurological problems. Moreover, since this is a mechanism that depends on the stabilization of that particular protein, we hypothesize that maybe that protein is a potential therapeutic target against situations in which there is excitotoxic insults like stroke or like other neurological disorders. For instance, if you look at this slide, There is neurons culture, right, treated with glutamate, which in excess can kill the cells because of the activation of glycolysis. But however, when you block the stabilization of PFB3, there is a normalization with the apoptotic rate, indicating that you need stabilization of PFKFB3 in order to get cell death. And these are a number of compounds that we are getting from a pharmaceutical company that are inhibitors of PFKFB3, and in culture and in vivo models of neurodegeneration are able to prevent the death of these neurons. So these are preclinical studies that hopefully will give us the possibility to provide a novel compound that properly administered into patients could be able to protect against neurodegeneration under these particular situations. So this is a very good example, and my point of view, in which it is important to understand basic mechanisms of the control of energy metabolism in the brain to therapeutics. And there are more examples, but there is obviously no time here to explain them all. So the question of, therefore, you have astrocytes in good collaboration with neurons. There is a crosstalk. There is a crosstalk. Astrocytes are mainly consuming glucose from the blood anaerobically, and neurons are consuming the astrocytic-derived lactate aerobically by the mitochondria. So these two cell types are specialized in performing completely different jobs in terms of energy. And there is a perfect coordination between both cell types in order to get the energy for neurotransmission, which is the important aim of all this process. So according to this, the mitochondria in the neurons needs to be very well specialized in consuming oxygen and these small molecules, bioenergetic molecules, because these cells depend on the mitochondria for survival or for energy production. However, in the astrocytes, which can survive perfectly with anaerobic glycolysis, they also have mitochondria. They also have mitochondria. What is the role of mitochondria in the astrocytes, therefore? What are the job of the mitochondria in the astrocytes if they virtually don't need them, at least for energy generation? So this is going to be the very last part of my talk. We have been trying to understand what is happening with the mitochondria. As you know, the mitochondria, amongst their functions, is the production of energy, of course. There are several other functions. But energy production is one of the most important ones. And energy production is conserved thanks to the occurrence of the mitochondrial respiratory chain, right? The mitochondrial respiratory chain. It's formed by several complexes which are placed in the inner mitochondrial membrane, and they are working together in collaboration by transporting electrons from the substrates in EDH, right, to oxygen, right? So there are several factors that are transporting the electrons, all right? And the work, this job of transporting electrons can conserve energy in the form of a proton gradient across the inner mitochondrial membrane, which is the source, the energy that you need to make ATP by the ATP synthase. That's clear. So, but there is one particular issue regarding this. It's not classically, the mitochondrial respiratory chain has been seen as the combination of several complexes that are embedded in the mitochondrial inner membrane and they are floating around, right? However, this concept is changing very recently, and instead of being separated complexes that needs to, find out how to send the electron from one to the other, there is very clear evidence that actually these complexes can be also joined and be together. They can be close assembled one to the other in different forms. For instance, you can have complex 1 bound to complex 3, or you can have complex 3 bound to complex 4, forming different supercomplexes. And also you can have complex 1, complex 3, and complex 4 all together. So this is important because when they are close together, the efficiency of the electron transport chain is much higher. So the efficiency where the electrons go from here to oxygen can be increased spectacularly. So this is not just a little change. It is a massive change, okay? So I'm going to go a little bit faster from here. So we examine what happened with the mitochondria in the astrocyte. This is what we wanted to see. And what we observe is that actually complex 1, right, is one of the most important complexes, obviously, is the respiratory chain. In astrocytes, right, is mostly free. It's not very well assembled to the other complexes of the respiratory chain. This is what this picture gives us. You don't have to understand this because I am explaining to you. This is a blue native gel electrophoresis. This is the way we have to see how are the complexes bound one to the other, okay? If anyone wants to know this, can come to my lab, and I will explain, or maybe this year, during biochemistry, I can teach them to you if you want, here. But this is important. When you compare to neurons, the proportion of free complex 1 in astrocytes is much higher than the proportion of free complex 1 in neurons. This is an important difference, right? We know that complex 1 is important. It's the complex that accepts electrons from NADH and transport them to ubiquinol to produce ubiquinol. Ubiquinol is then taken by complex 3 in the respiratory chain. However, complex 1 is also an important source of superoxide. Because these electrons, besides going to ubiquinol, there is a specific site in this complex where oxygen binds. And this part of these electrons can be directly transported to this oxygen, forming this species, which is superoxide anion, which is a free radical species or a reactive oxygen species in the mitochondria, that then it can be converted to hydrogen peroxide. So actually complex 1, besides transporting electrons, is also an important source of superoxide and free radicals. In fact, when you go and measure the production of hydrogen peroxide in neurons and astrocytes, which is here, can you see astrocytes produce a lot of hydrogen peroxide when you compare to neuron, in accordance with the higher proportion of free complex 1. So having free complex 1 is important to produce hydrogen peroxide. So in essence, the message of this is that there are two different metabolic shapes between neurons and astrocytes, not only in the way they consume glucose. Astrocytes glycolytically, neurons actually take lactate from the astrocytes to produce aerobically energy, but there is also specific differences in how they organize the respiratory chain. In the neurons, here in the left, most of the complex 1 is bound to complex 3, so there is a very close relationship and assembly between the complexes, which makes the respiratory chain very highly efficient, very highly efficient. Whereas in the astrocyte, there is a proportion of complex 1 which is separated from complex 3, and therefore the efficiency of the respiratory chain in astrocytes is lower. However, that gives the opportunity to astrocytes to produce much more reactive oxygen species, right? Why is that? I mean, the more the electrons in the neurons that you have that are perfectly assembled to the respiratory chain, the less the chances you have from these electrons to go to oxygen, all right? Whereas in the astrocyte, because you have very little electrons go through the respiratory chain, you have more options of these electrons to go through complex 1 to oxygen. So this is a very important difference. So we are now investigating what is the significance of mitochondrial ROS in astrocytes. And I had just 2 slides to show you that we have preliminary data with that, in which we have made a mice, genetically modified mice, that is able to reduce the mitochondrial reactive oxygen species production, specifically in the mitochondria, in astrocytes. This is just a normal, usual, traditional genetic approach to do that, okay? And you can measure, actually, right? And if you go that and see what happened to the animal, you see that they have, these animals, when you examine and you explore their behavior, they have signs of loss of short-term memory. We have studied also the whole mechanism responsible for that, but I have no time to describe it. So, therefore, understanding molecular mechanisms controlling energy metabolism in the brain is not also important for our knowledge on how are we working. It's also important to identify specific therapeutic targets. It's also important to understand how these brain cells manage the production of reactive oxygen species, which is very important and very present in the pharmacy, therapeutics, because of the massive use of antioxidants in an indiscriminate way. So one has to be careful when one wants to administer antioxidants to the brain, because it is, according to this data, important that the astrocytes produce a lot of ROS, because if you remove them by this genetic system, we know that there is a loss in memory, short memory formation. Okay, so one has to be careful with the use of antioxidants. So in conclusion, just 3 main conclusions from this talk. We believe that, 3 points that you home take messages, okay? Glycolysis is highly active in astrocytes due to a highly active proglycolytic enzyme, PFKSB3, to support oxidizable lactate to neurons, okay? This is important because also we identify PFKSB3 a potential therapeutic target for the reason I explained to you before. Second take-home message, the respiratory chain, the oxidative phosphorylation, OXPHOS, okay, is differentially organized and adapted to astrocytic and neuronal metabolic shapes. And that is not only that it's adapted to the energy conservation, but as a consequence of that, they have different production of reactive oxygen species. In the neurons, because they have an efficient electron transport, they have very low ROS production under basal conditions. However, under physiological conditions, astrocytes are inefficient. The mitochondria is inefficient. However, that allows these cells to produce high amounts of ROS. And this is under physiological conditions. This is not pathological conditions. And finally, we observe that the physiological role, or one physiological role of mitochondrial reactive oxygen species produced by astrocytes, physiologically is modulating metabolism. I didn't show the data, but it is. Neuronal function and behavior. So therefore, careful with antioxidants that remove mitochondrial ROS, right? Careful with that, because you can interfere with your memory. And finally, this is a picture with, I know this is a little bit old, part of my group here in the Institute of Functional Biology and Genomics, just close to the INCIL. And this is the name of the main collaborators. One of them is here, Giovanni Marsicano. And this is the funding. So thank you very much. I'm sorry that we started 5 minutes late. So if you want to make a question, if not, if you want, we can wait for the end of the symposium, and if you have a specific question to me, you can make it to me later, okay? In order to be on time. But anyway, if you have a question now, you can do it. Thank you. Conéctame este. Sí, thank you. Anybody? You want to? Okay, yeah, go, yeah. Do we have a microphone for the people?
Can I speak loud enough?
No. But I think it's better to have the microphone so the people around can hear the question, because I'm sure it's going to be an important question.
Very nice and exciting. So the idea is astrocytes make more ROS. Neurons are more protected by antioxidants that they produce themselves. So you would expect that the astrocytes have a way to handle those ROS species. And is there anything known about that?
Yeah, that is an important question, indeed, yeah, as I expected. It's not that the neurons, they have a better defense against ROS. It's that they produce less ROS because their mitochondria is very efficient. Yeah. However, astrocytes, because they produce a lot of ROS, they also have a very important endogenous antioxidant systems. So this is, one thing is the rate at which they produce ROS, and another thing is the rate at which they scavenge ROS. And actually, astrocytes have a very potent and robust antioxidant system that they also give to the neurons to protect against insults and so on. So the redox metabolism between neurons and astrocytes is another very fascinating topic that I am interested to, but you know, would require more time to understand it. Thank you. Okay. Oh yeah, a question there.
I have sort of a clinical question back to the PF-KFB3.
Yeah.
So I understand how in a generalized pathologic state like status epilepticus, where it requires lots of glucose to keep the brain alive, that could work as a therapeutic agent. But you mentioned stroke specifically with that. So that uses glucose, but that's so specific of an area. How can that relate to sort of the same thing as this generalized pathologic state like status epilepticus?
In a stroke, you're talking about, because I didn't get very well the question. Okay, sorry. Yeah.
I know it's hard to hear down there.
Yeah.
So you mentioned that the PF-KFB3 would be helpful in stroke, or could be helpful in stroke, in something like stroke, because it would block that energy pathway and allow the brain to maintain its glucose. But stroke is very specific, lack of oxygen, not so much glucose. But I can understand how it would be very helpful in a generalized pathologic state like status epilepticus. I just was wondering how, because stroke is so specific, that could be a good agent.
Okay, so your question is, how you don't see very clear why inhibiting a glycolytic enzyme could be protective in a pathological situation like a stroke? This is your question? Because I'm sorry, but I didn't.
We can talk about it afterwards.
Yeah. I think that it's very interesting, the question, I'm sure, but it didn't get exactly your point, all right? But I'm sure that we can discuss about that later. So I think that, thank you very much. Can we shoot to the next? Thank you.
Good morning, everyone. It's a great pleasure to be here in Salamanca and in this fascinating meeting, and I want to thank Juan Pedro for the kind invitation. He has talked about astrocytes and neurons, and indeed, in thinking about brain function, every one of us just thinks about, most of the time, neurons and circuits. I would like to tell you about two other cell types in the brain that are less well known and which have been neglected for many, many years, almost 100 years, thinking that brain function resides in neuronal circuitry. And these two cell types that I'm going to talk about are neural stem cells here and microglial cells. And these cells, we are starting to know more about them, and they're fascinating because they are being generated very early in fetal development, both of the types. Neural stem cells produce all the neurons that we are born with, and they continue to inhabit our brains in two specific locations, giving rise to neurons throughout our entire life, even in humans. Microglial cells are a type of immune cell that resides in the brain. They get generated very, very early in development from a hematopoietic organ, a fetal organ. They then colonize the brain and remain there for our entire life. And they are a sort of tissue-specific macrophage. So they are an immune cell, but they are self-renewing. They can renovate themselves, regenerate themselves. They have a function during disease, and most of you know that microglial cells are the key cells for neuroinflammation, and that has a lot of relevance for different diseases in the brain, including neurodegeneration. But these cells that are there in the brain from very early on in fetal development are highly plastic, and they perform functions that are related to physiological function. And we don't know yet exactly how they do so, but they are main contributors also to brain function and well-being. So we study in the lab neural stem cells, and neural stem cells are just an example of adult stem cells that we have in our tissues that have the capacity of self-renewal to give rise to more of themselves and be maintained with all their properties, while they give rise to progeny. In the brain, they give rise to oligodendrocytes, neurons, and astrocytes. And mostly in the mature brain, they just produce new neurons. That contributes to tissue renewal. And understanding their function under physiological conditions is important because these cells, maybe we could convince them to repair if we know how to activate them. The endogenous populations could be used for repair by therapeutical pharmacological intervention. Now, they could also be a stem cell source to increase their size or expand them ex vivo, and then try to make neurons in vitro for transplantation. It's also important to understand that we consider that the loss of potential of these cells may underlie aging phenotypes, the deterioration of our tissues, including our brain, and also that mutations in these cells can produce cancer, glioma in the case of the brain, and understanding, therefore, their deregulation may be important also for tumor treatment. One way to understand how these stem cells behave is by looking at the effect of their microenvironment in these cells, how they talk to the cells that are surrounding them. This is what we call niche biology, and it's exactly what my lab has been working on for many years now, understanding how the microenvironment of these cells regulate their behavior. Now, the concept of a niche, of a microenvironment for stem cells was proposed 4 years ago by Raymond Scofield as an environment to explain the unlimited proliferation and failure to mature of stem cells. Now, if we think about the stem cells that we have in our brain, those neural stem cells give rise to other cells that have a reduced potential compared to them, which are neural progenitor cells, and these cells divide a few times and differentiate into neuroblasts and neurons. So they are surrounded by many different other cell types, astrocytes. Juan was already telling you about astrocytes, neurons, microglia, endothelial cells of the blood vessels, oligodendrocytes, etc. So, as I was telling you, we have described over the years many different signaling pathways that triggered by molecules produced by these different cell types in the regulation of the state of these stem cells under activation. But I would like to here highlight a different concept that's emerging strongly in the field. And if one goes back to the postulates of the Scofield proposal of a stem cell niche, and I want to stress out the fact that this was 40 years ago. Aside from these different postulates that that are listed here, this second one in bold, it's extremely interesting from the point of view of nowadays. And it's the niche. He postulated the niche should integrate signals reflecting tissue and organismal state. What comes down to the point from this statement is this question: are stem cells in any part of the body capable of responding to signals or situations in other part of the body? can our neural stem cells sense changes in the rest of the body? So the idea is that that's why I'm proposing to you to try to convince you that we can talk about an extended niche, as Scofield first postulated. This would mean that there is some sort of remote signaling. Stem cells not only respond to short-range signals in their microenvironment, but also to long-range signals from other parts of the body that come from other parts of the body. But I want to remind you, and I don't need to say that in this audience, because this is one of the problems that pharmacy faces when treating the brain, that these cells are, like neurons and astrocytes, behind a blood-brain barrier. So if they can be sensing remote signals, we have to take into account that they have to cross somehow the blood-brain barrier. Now, these cells, that we can isolate them from the brain, and we are quite efficient in getting nice cultures of neural stem cells. So these cells that we study reside in the wall of the lateral ventricle. This is mouse tissue, and we can dissociate them, seed them as single cells in a convenient medium, growth medium, and then we obtain these clonal aggregates which are called neurospheres that can be expanded for many passages, and each clone was generated by a single cell that we call neural stem cell. Now, if we differentiate a clone, we see that cell that initiated the neurosphere has generated astrocytes, oligodendrocytes, and neurons, so that cell was multi-potential. But the problem in the field has been that we don't know exactly what's the relationship between the nature of the neural stem cell in vivo and the nature of the cell that generates a neurosphere in vitro. And the problem was that we didn't have markers to prospectively isolate cells that we identify in vivo and then assay them in the culture plate, as hematologists do with the hematopoietic stem cells. So in this context, Jose Manuel and Germán decided to try to set up a method to isolate different populations from this structure, from this part of the brain, and classify the cells that we obtain in different fractions that we could assay functionally. And I want to go quickly through this because, as you can see here, we have put like what we thought could be 3 flavors of neural stem cells. You have the neural stem cell, now you have 3 versions in different colors, because we thought that maybe that procedure would allow us to see whether we have different states or different cell subtypes of stem cells. With that in mind, we excluded, I'm going to go quickly through this because this is only the strategy that we have used using fluorescent activated cell sorting using flow cytometry. And this is with the tissue, we dissociated the tissue, did negative selection for microglia, endothelial cells, erythrocytes, and oligodendrocytes. And with these lineage negative populations, we used different markers that have been used in the literature, and some of them have come from our laboratory to fractionate that cell pool into different populations. And if we do that and apply other markers to separate neural stem cells from astrocytes or neural stem cells that are activated because they respond to mitogen or are not activated because they do not respond to this mitogen, as I was telling you, I don't want to go into the details of this, we come to this sort of classification. So we can fractionate this tissue, and this is the important part, using surface markers into 8 different cell fractions, okay? And with these different cell fractions, for some of them we know exactly what they are astrocytes, but we could also get 3 fractions. We could isolate what we propose to be neural stem cells in 3 fractions. Now, this is not a demonstration that we have these populations and we have to assay them, but we have our entire tissue homogenate classified into 8 classes. What we did with that is to do an RNA-seq analysis of all these populations. So we have thousands of genes, and then you do a bioinformatics analysis to see whether the transcriptomes are coherent and how the different transcriptomes are related across the 8 cell fractions.
And this is something that bioinformaticians can do. They can tell you how the transcriptomes group together and how they are ordered in a progression, because they can see how the gene expression is changing throughout the different cell populations. What you see here is the pseudo-time ordering of these populations. Each dot is one different independent isolation. And you can see that we have grouped here the different isolations for a group of what we call neural stem cells, another one, and another one. We have 3 groups, and then the neural progenitor cells, the neuroblasts, and the cells that we cannot classify in any of the groups. So the idea was that the expression profile, the molecular signature is coherent in these groups, and we can order them. How do they behave in vitro? So we put them in culture. And instead of showing you now the grown neurospheres, let's take a look at what I'm going to concentrate only now in these 3 fractions, these 3 classes. How they behave, not when they have formed the neurospheres, but when we plate them in a single cells 24 hours later and 48 hours later. And what you see here is the first group never divided in vitro, never. And they disappear by 48 hours. The third group divided really fast. At 24 hours, we could see, you see here 2 cells, and here we have 4 cells, and they start in 8 days, in 12 days later, we have these perfectly grown neurospheres. Now this other fraction start in a delayed manner, so it takes them longer to initiate division, but they finally make neurospheres that can be expanded. So we can propose that by their in vitro behavior, these cells would be, this is compatible with cells being quiescent in the first group, and the last group activated neural stem cells, and the group in the middle, what we call now in the field primed stem cells. And this has been shown in the muscle. In the muscle, in the skeletal muscle, stem cells never enter, they are never activated during lifetime unless you injure the muscle. So if you you stop the muscle with a needle, the satellite stem cells enter activation, and they try to produce new muscle fibers. And this happens in the muscle. But if you you make a lesion to one muscle, now we know that the muscle in the contralateral leg or hind limb, the cells there become alerted, which means they are already prone to activation, but they do not enter activation. And when the lesion is restored, they go back to quiescent. So in the neural stem, in the brain, we also have quiescent cells, primed or alerted cells that react to something, and then if nothing happens, they go back to quiescent state, and fully activated cells that divide and produce new neurons. The problem here is that, as you can see, if we pay attention to these, quiescent cells are never in vitro. So when we culture neurosphere, we never have a quiescent cell in the culture plate. So we cannot even analyze their function or how they behave in vitro, right? So to show you that these cells are really what we think they are, we did an in vivo proliferation study. We can give the mouse EDU to label proliferating cells, and we can give them EDU, and 1 hour later do the same analysis that I just showed you. And one can see that quiescent cells they do not divide, they do not incorporate EDU, but activated neural stem cells do. So this is, again, okay. If we do more pulses of EDU to try to catch the cells that can proliferate, we start seeing that we can get the primed cells because they are almost there. They are prone to divide. So we can catch them with consecutive pulses of EDU, but we never label the quiescent cells. So quiescent tendencies are highly refractory to cell division, even in vivo, and we cannot culture them in vitro. So we decided, instead of showing you this graph, I'm going to show you the same graph in this format. Okay, so this is the same result that you just saw. But imagine we label these activated stem cells, most of them are activated with these pulses of EDU, and now we wait 21 days to do the experiment. What happens is that the cells that keep dividing, well, what happens is that then most of the cells that are EDU positive are the quiescent cells. What this means is that neural stem cells in the brain that are activated and therefore they become labeled with EDU, they do whatever they have to do, they produce neurons, and at some point they return to a quiescent state. And this has been highly debated in the field, and this is for therapeutical purposes. If we want to activate neural stem cells in the brain, there was a big concern in the field because most people, some results have indicated that if you activate the neural stem cells, you are going to accelerate their exhaustion. So they become activated and they become exhausted. And there were no real proof that these cells, when they are activated, can return to quiescent, which means that if we activate them properly, they can return to quiescent and we will not lose our neural stem cells just by activating them. So this is good news, and this gives us a way of analyzing this back to quiescence process of these cells. So this gene expression analysis that we did allow us to do something else. And again, bioinformatics analysis can tell us what are which genes are being transcribed that tell you something about the function of those cells or about what's happening in those cells. And when one does this sophisticated analysis, what we saw is that quiescent cells, if we focus exclusively on the quiescent cells, have increased activation of genes that are related to the response to inflammation. And I presented to you before the microglial cells, right? So we go to inflammation now. What is known about inflammation and neurogenesis, the production of new neurons in an adult brain? So the general agreement in the field is that inflammation is bad, as we think it is for any situation in the brain. So neuroinflammation is bad, and this is a negative. Because what people do when they increase inflammation is that neurogenesis becomes reduced, okay? But if you look at the role of inflammatory cytokines in zebrafish, which is an animal that regenerates, has normal regeneration, then those cytokines have positive effects on the neural stem cells in the brains of these fishes. So we hypothesized that maybe in stem cells you have positive effects of inflammation that we don't see because we are only seeing the detrimental effects. So in order to produce neuroinflammation, what we did was what most people do experimentally is to inject animals with an endotoxin, which is a lipopolysaccharide of the bacterial cell wall of gram-negative bacteria intraperitoneally. So we are injecting it IP, and this is a potent activator of the innate immune system. And you can see as here, as has been described, many genes are downregulated after LPS injection. But there are some genes that we know now, because of our studies, that are associated to neural stem cells and not other cell derivatives or cells in the brain, are upregulated. So maybe we are having like the yin-yang of the neuroinflammation, good and bad effects to the neural stem cells. So if let's apply then, after LPS, this EDU experimental paradigms. And this may look a little complicated, but the idea is that if we inject LPS, what we see is an activation of the neural stem cells. And this activation is transient. What you're seeing here is that for the activated cells at 6 hours, 12 hours, and 24 hours after the LPS injection, we can see that they incorporate EDU at first, and then that decreases. And quiescent cells do not respond to LPS. Now, if we now, instead of doing these injections, inject the animals several times and then wait for 12 hours to see what happens with the cells that incorporated the EDU, we find that quiescent cells, we have a higher proportion of quiescent cells that are now EDU positive. This experiment means that when we inject LPS, the neural stem cells become activated, and 12 hours later, they go back to a quiescent situation, so there's a cycle, okay? Of course, because of this cycle, we can, I'm not going to show it to you, but what happens is that we have a decrease in the progeny. So LPS reduces the production of neurons because the response to that stimulus is activation and back to quiescence. So neural stem cells are responding to this in a natural way, in maybe trying to keep the quiescent pool of the neural stem cells. Okay, so I'm going to do that faster. What LPS does is activate toll-like receptors in macrophages that are in these specific locations in the brain where there is no blood-brain barrier. And then these cells produce TNF-alpha that activates microglia throughout the brain. And we can see the profile of the TNF-alpha production in the body and in the brain. Doesn't matter. We can demonstrate that this TNF-alpha is produced by microglia, no matter, don't worry about the graph. And this TNF-alpha has never been related to a quiescent phenotype. So it activates, it activates a death receptor in the membrane of the cells. It's a pro-inflammatory cytokine that normally causes, in many cases, causes cell death, apoptosis, but can also activate the NF-kappa B pathway that regulates transcription of genes that are needed for cell survival and other functions. So we put this TNF-alpha thinking that maybe the LPS signaling at the end is a signaling of TNF-alpha. We added this to the neurosphere cultures, and we could see, after growing our neurospheres with TNF-alpha, that we were observing cytostatic effects. Like the idea is that several days, 5 days later, we could see more cells that had never initiated the neurosphere formation, so we're still as single cells, single viable cells. This is a new function for TNF-alpha. It has never been shown that it can act as a cytostatic molecule. So is this so in vivo? So this is the same experiment that you just saw, showing you that with LPS, neural stem cells become activated and then they go back to quiescence. What happens if we eliminate TNF-alpha signaling by using mice that are deficient in the receptor 1 for TNF-alpha? Does that not happen? Okay, so the idea is, in thinking about inflammation, especially we have to start distinguishing between chronic inflammation associated to many diseases, or many injuries and diseases in the brain. That's okay. But there is probably, let's say, many of the molecules and mechanisms that have evolved in inflammation were there for repair and for regulation of of stem cells. And those mechanisms are good. So we have to, it's the same argument that Juan was giving you as a take-home message, is that we cannot just say inflammation is bad, because maybe we need part of the signaling mechanism of what we call inflammation or innate immunity to promote or preserve the capacity of stem cells for repair. And this is from an evolutionary point of view, and this is my last slide. Innate immunity is very ancient. All the animals have innate immunity. That's the basic first response. Microglial cells are part of this innate immune system. And then we have in mammals, adult mammals, not fetal, adult mammals have adaptive immunity. And what adaptive immunity is a very efficient mechanism. In homeothermic animals, we have to cope with infection because at 37 degrees pathogen can kill the organism. So we have to evolution has favored the wound healing over regeneration. So we have lost regenerative capacity, because of this sophisticated immune response, but we still have this innate immunity that in other animals is the base for regeneration. So if we understand what innate immunity does to stem cells, which are maybe the equivalents to cells that in other animals are the basis for epimorphic regeneration, maybe we can help our tissues to respond better and to repair or regenerate in our bodies. So this is my group. I have mentioned the work of José Manuel Morante, Germán Belenguer, Pere Duarte, and Tony Jordan has been the person in the lab who has done the bioinformatics analysis. And I want to thank you all for your attention.
Okay, yeah, Ruben. There is time for 1 or 2 questions because we want to keep very tight on time.
I'll try to make it short. President Borges is so you're saying that TNF-alpha is acting on 2 pathways. So one's escape, the other one's
NF-kappa B.
Ruben, can you wait 1 second to have the microphone so everybody can... No, it doesn't matter. You don't need to go that fast.
Okay, so 2 pathways, right? Caspase and there should be a balance there. So you were blocking the TNF-alpha, the whole thing. But what happens if you block only the caspase? Can you improve quiescence, coming back to quiescence?
We are doing that experiments right now. We have done many other things. I mean, because there is another receptor for TNF-alpha, which is ARC2, which we now know that it's involved in the priming of the cells. So it's more complicated even than what I've showed you. What for us is highly interesting is that we have not, in line with the question, we are now using inhibitors of NF-kappa B. And the thing that we're trying to do is, because we have never been able, us or nobody else on the planet, culture quiescent neural stem cells, put them in vitro. We are going to inhibit NF-kappa B to see whether we can have finally a quiescent stem cell in vitro.
Looking forward.
Thank you.
So thank you very much. I think that I have 1 question for you, but I'm not going to tell you now. I will talk to you later, and has to do with the possible role of the anti-inflammatories, therapeutics in adults, and whether there is any evidence that it's affecting the neurogenesis in humans. But we can talk about that later because of the, we are tight on time.
But it's pretty much like the thing you were saying with antioxidants.
Yeah, I can imagine. Thank you very much. So now, Professor Giovanni Marsicano. Whenever you like.
Hi, everybody. Thank you, Juan Pedro, for inviting me. Thank you for organizing this meeting. They told me not to panic if the slides were not appearing. I'm not panicking. Does it start? So I have to do myself. Huh?
No, it's already. Oh, you want to check? I will do it.
Where is it?
No. It's better this way. Okay, yeah. Do it.
I thought that it was automatic.
Doesn't matter. No. I thought also that it was, but it wasn't.
He told me, don't worry if it takes time.
Is that one?
Okay, it's this one.
You have to. No, it will come, don't worry. It takes
okay.
a few, it will come.
So, did I do something wrong?
I don't know why. Okay, that's that, okay. Don't worry.
I know. You are okay? I mean, we can talk about football if you want. No, better not. I'm Italian, actually. Okay, good. So today I would like to speak to you a little bit about CB1 receptor 7 endocannabinoid system. As you can see from the name, CB1 cannabinoid receptors are coming from this plant Cannabis sativa, marijuana that is known since many years by humans to have effects on the biology. And then, but we didn't know actually how the plant was acting on the cells, and until, let's say, middle of the last century, when the chemical compounds of the plant were identified, and there are actually almost 100 specific compounds of the plant. Most of them are biologically active. For most of them, we don't know how they work, but for sure the most important, the most famous is delta-9 tetrahydrocannabinol, which is the main responsible of the psychoactive effects of the drug. When we knew what are the compounds of the plant, we didn't know how they act on the plant, and this is important also for the rest of the talk. There were many different theories, hypotheses, and so on. However, until the late 80s, the dates are quite important, so keep in mind, when actually 7 transmembrane G protein coupled receptors were identified to respond to these compounds like THC or synthetic cannabinoids. So at this point, there was a kind of family of exogenous ligands and an endogenous receptor. Of course, what was missing was the, why we have a receptor. Maybe evolution gave it to us to enjoy cannabis, which is a possibility, but maybe most probably the receptors are endogenously activated by some compound. And indeed, after a couple of years, the first compounds were identified as lipids in the body, in the brain, and this is important, they are also lipids, like most of the exogenous cannabinoids. So cannabinoid receptors are mainly lipid receptors. So at this point, we knew that there was an endogenous cannabinoid system, endocannabinoid system, which is involved in many functions. This is just a limited list of functions in the brain. However, if you go to the literature, you start to see this, and you realize that basically there is nothing that happens in the body that at some point will not involve endocannabinoid signaling. So the question of my lab is basically, how can CB1 receptors do all these beautiful things? And this is a very simple question. Okay, if we go to the textbook, you see that CB1 is of course considered a G protein coupled receptor on the plasma membrane, and when it is activated by exogenous or endogenous ligands, normally CB1 is coupled to GO inhibitory G proteins. Not always. This is really the oversimplified system. However, this is leading to activation of potassium channels. CB1 can inhibit directly calcium channels and opening other potassium channels. If you remember from physiology, if you have an excitable membrane, if you open to potassium and close to calcium, what happens is that you have a neuronal hyperpolarization, and if it is a neuron, you have an hyperpolarization of the membrane, and you have a decrease of excitability of the membrane. On top of that, CB1 is linked to the activation of many intracellular pathways, for example, MAP kinase, but many others, so making already the textbook, the simplified version of a system already very complex. The other side of the endocannabinoid system are the endocannabinoids. As I told you, most of them are lipids. So they act as neurotransmitters. Now we know that they act as neurotransmitters in the brain, but of course they are different from classical neurotransmitters because they are not water soluble, so they are not stored in vesicles. You know, glutamate, GABA, acetylcholine, whatever neurotransmitter, we know very well that they are stored in vesicles, and then they are released by fusing the vesicle to the presynaptic site, and this is releasing in the water extracellular space the water-soluble neurotransmitter. This cannot happen with endocannabinoids because they are lipids. So the idea that we have is that they are directly produced by membranes. And by membranes, I mean all membranes of the cells, not only the plasma membrane. So the mechanism is it's quite complex and depends on the compound, but it follows more or less always the same scheme. You have a stimulus of a membrane, but can be a depolarization or whatever, that is leading somehow to an increase of calcium, or you can have also direct activation of this enzyme 1, let's call it, by other pathways. What happens is that you take arachidonic acid and membrane precursors, And then this first enzyme is fusing these precursors with arachidonic acids, making a second precursor, and then you have a second enzyme, and then you have a production of your endocannabinoids, which as lipids can very well move in the membrane. This is also important. They move laterally on membranes. This is now pretty much shown. But however, they can also, through mechanisms that we still do not understand, can be released from the cells. Then they can act on CB1, either on the same cell or in an adjacent cell, and exert all the functions that I told you before. And then they are rapidly uptaken and degraded. So what is important in all this is that this can be very fast. So the idea is that mainly the endocannabinoid system works in a on-demand, in a phasic way, and not in a tonic way. Actually, now there is growing evidence that endocannabinoids can work also in a tonic way, but this is another story. Okay, so this can be very fast. In electrophysiology, for example, they can measure this in few tenths of milliseconds. You can have stimulus, production, action, and degradation, okay? And so what in the brain, what do they do with endocannabinoids? They do a lot of things, but this is probably the best characterized. So if you imagine here to have a presynaptic membrane and a postsynaptic membrane, so this is a synapse, we know very well that when the signaling from the presynaptic to the postsynaptic cell is going through an action potential that is coming to the presynaptic side. This is triggering all a cascade at the presynapse, and this is fusing the membrane of the vesicles to the membrane of the presynaptic side and releasing the neurotransmitter, which is then activating postsynaptic cell. Already in the early 90s of the last century, there were some physiologists that identified the possibility that that there was some communication that was going retrogradely, okay? But at that time, these people were kind of not considered very well because I read reviews in which there were really fights about that because they were saying, okay, we know that neurotransmission is through vesicles. In the postsynaptic side, there is no vesicles. So what is this retrograde signaling that you are seeing? So they were accused to have artifacts in their experiments. Instead, in 2001, 3 papers at the same time, 2 in Neuron and 1 in Nature, showed that actually what happens is, in many cases, is that if you have activation of the postsynaptic side, what you do is simply that you produce endocannabinoids that somehow travel the synaptic cleft, bind to CB1 that is generally presynaptic, decrease calcium, as I told you before, decrease membrane excitability, and this is leading to decrease of neurotransmitter release. So this is known as retrograde suppression of neurotransmitter release. It's very well characterized. Now it's present at basically everywhere in the brain and even in the peripheral nervous system. So keep in mind, CB1, the main effect is to decrease the release of neurotransmitter. So at this point, it becomes important which neurotransmitter CB1 is regulating. And so to answer this question, we have to know where is CB1, on which type of cells. So first of all, you see that CB1 is very highly expressed in the brain. It is actually, I forgot to say that the plant cannabis sativa and cannabinoids have a lot of records. So they are basically, cannabis has been the first plant that has been cultivated for reasons independent of food intake, that you are not cultivating to eat, but to cure things or to make other stuff. And then it is the first holy plant that was used in India to communicate with gods. And another record that this system has is that CB1 is probably the highest G protein coupled receptor expressed in the brain. There is really tons of that. There is no competition with opioid receptors, dopamine receptors, no way. In terms of quantity, CB1 is absolutely more abundant. Even, we will see later that this might be not so important. Okay, there is a lot of CB1 in the brain, and if you go in a particular region, but this is true throughout the brain, in the hippocampus is very nice. You know, the hippocampus is a brain region that is very important for memory, and it is very interesting for physiology and anatomy because it has a very well-defined distribution of cell types in the structure. And what happens, if you observe CB1 expression, this is mRNA, so what you see is the cell body of the cells. And you see that CB1 is very, very, very highly expressed. This is an insane amount of expression in these cells that are GABAergic interneurons. So they are inhibitory neurons in the circuits of the hippocampus. However, CB1 is present even if at much lower levels in these cells that form this line around, that are the CA1 and CA3 pyramidal glutamatergic excitatory cells, and therefore CB1 could be also regulating excitatory principal circuits. So you can see already that you can have an inhibition of excitation, inhibition of inhibition, and if we don't know anything about how the brain works, the only thing that we are pretty much sure is that everything works through increased inhibition, increased excitation, or decreased inhibition and decreased excitation. It's always a balance between plus, minus, and I'm talking to you as a result of this. And so one question that we had at the beginning is, are there cell type specific functions of CB1? Because you can imagine, you decrease inhibition, it's completely different than decreasing excitation. It's exactly the opposite. So what we did already many years ago, we generated mice lacking CB1 in specific cell population. Here I go a little bit quicker, but we have these mice that we call GLU CB1 knockout and GABA CB1 knockout. If you look in the hippocampus here, you see again the expression of CB1 in these strong dots that are the GABAergic interneurons, and then you have this line that are the glutamatergic neurons. I think you know that GABA is the main inhibitor in neurotransmitters and glutamate is the main excitatory neurotransmitter. And if you delete CB1 in cortical glutamatergic neurons, you do not see any more the expression of CB1 in the CA1, CA3 line of cells, but you still see the strong dots. In what we call the GABA CB1 knockout mice, you find that you see only the line of principal cells, but not the GABAergic interneurons. So we used these mice for many things. One important thing is the expression of protein. So if you check here the expression of a protein that is presynaptic, I told you, you see that you delete CB1 in glutamatergic cells, basically you do not see the difference with the wild type. So that means that we deleted a small amount of the protein in the brain, okay? Instead, if we check the mice that have a deletion of CB1 in GABAergic cells, it's not a full knockout because you see the full knockout is like that, and here you still see a faint staining, but you lost a large majority of CB1 in the brain. So that means that GABAergic neurons contain the large majority of CB1 in the brain, and cortical glutamatergic neurons contain a minority of CB1 in the brain. This is important. However, then we wanted to know from a functional point of view. We made many, many experiments. I just showing you one example. And this is a very simple experiment of fasting-induced food intake. Who made ever the experience of cannabis knows that one of the first things that you do when you smoke some joint is that you go to the fridge to eat. So it is very well known that the endocannabinoid system and CB1 receptors are promoting food intake. And this was shown in, a paper in Nature experimentally that CB1 knockout mice, after fasting, eat much less. This was shown in 2001. And we did the same experiments with a full CB1 knockout, but also with our conditional mutants. So the idea is very simple. You fast the animals 24 hours, and then you check how much they eat when you refeed them for 1 or 2 hours. So what you find is that mice that do not have CB1 eat less than the wild type. Okay, we reproduced the results of Di Marzo. But then you check the GLU CB1 knockout mice that lost a little amount of CB1, and what you find is that the phenotype is very similar to the full CB1 knockout. So it's this tiny amount of CB1 that is responsible for the general effect of the system. And this was already a surprise, but even was more surprise to check these mice, where we lost the great majority of CB1, and what happens is that these mice eat more. So the idea is that basically there is a bimodal control of food intake by the endocannabinoid system, and what is interesting here is that it's the same system that is making you eat when you are hungry, that is also limiting the amount of it, that you eat, because it could be dangerous, probably. So it's the same system that can play on both the situations. So a conclusion of this is that quantity, at least for CB1 receptors, but I think in general in life, is quantity is not quality. So a tiny amount of CB1 is responsible for the whole effect of the endocannabinoid system, and the larger amount instead is just a break. It's a system that is doing the contrary. Okay? Cool. So this is a conclusion one. I'm already very late. And CB1 is acting as a classic GPCR regulating behavior. The CB1 functions and signaling are not intrinsically by emerging from cellular location, and et cetera, et cetera. This is not what I said, but this is coming from all the studies that we did. But there is a question. Is that all? So, discussing with friends years ago in the field, we were saying, but when you make an immunosource CB1, you see a lot of CB1 inside the cells. What is it? And the main interpretation was CB1 is trafficked from the Golgi and so on to go to the nucleus, and therefore you find it inside the cell, which is obviously true. However, CB1 ligands are lipids. And as I told you before, lipids can move very easily within membranes, and all the membranes of the cells are connected to each other. So lipids can go inside the cells very easily. And then, if you remember the dates I told you before, between basically THC became famous in 64, and then the receptor was identified in 1990. In between, what is there? 68 and 70s, right? And so what we call the 70s, the hippie scientists at work, that were trying to understand how THC works in the brain and in the body, and basically anybody who had a biological system was throwing some THC on top just to see what happens, because this was cool. And you have very nice, very interesting papers and very interesting hypotheses, and you find papers, for example, in 71, in 72 and 76, in which people showed that cannabinoids can have very important effects on mitochondrial functions. Okay? Of course, when 20 years later they found that there is a receptor, these data were kind of dismissed as unspecific in the sense, and this makes a lot of sense also, because mitochondrial membranes are very sensitive to lipid composition. So what we were saying, this is unspecific because cannabinoids are lipids, they alter the membrane composition, and therefore you have effect on mitochondria. However, this was in the background of my brain, and one day I met Pedro Grandes in Bilbao, that is a neuroanatomist that you might know, and I asked him, did you ever see CB1 in mitochondria? And when he's doing immunohistochemistry in electron microscopy of CB1, and his answer was yes, but of course it is background, because GPCRs cannot be in mitochondria. This doesn't make sense biologically. And I told him, okay, probably it's true, it's background, but I send you some knockout and you tell me what happens. And after several months, because electron microscopy experiments are very long, he sent me these pictures, and you see that here you have a very nice staining. You see CB1, a lot of CB1 on the terminal. Here, this is probably a GABAergic interneuron. And then you see the mitochondria, you have 1, 2, and you have 1, 2 here and a lot around. And then if you go to the knockout, basically you find some background. But because of course you don't have, for example, here or here and so on, but it's not specifically more in mitochondria than anywhere else. So we think that this is, we had the idea that this could be really a specific staining, the specific presence of CB1 on mitochondria. We quantified this, and we know that now that around 10–15% of total CB1 in the brain is in mitochondria, and then you can make a Western blotting, and you can identify CB1 in purified mitochondria. Okay, So we called this MTCB1. Giovanni Benar made a lot of experiments. I show you just one, in which he gave a CB1 agonist to an isolated population of mitochondria from the brain and found a decrease of respiration, okay? So CB1 activation decreased respiration, and this is not present in CB1 knockout. So this is cannabinoids decrease respiration, but this is CB1 dependent. If we increase, depend on the absolute doses depends on the preparation, but if we increase the dose, at some point, we will have an effect also in the knockout, which is probably due to membrane interference. But however, until up to a certain level, we have a specific receptor-mediated effect of cannabinoids on mitochondrial respiration. So this is the idea is that MTCB1 signaling directly regulates mitochondrial functions. This gave us some good papers and good covers. This is a very nice picture. This is a mitochondrion, right? You recognize very well. But the question was, what is MTCB1 good for? And this is not an easy question, because basically what you have to do is to select the receptor in the same cell in two subcellular positions, different. So it's not an easy case. Using pharmacological tools that can go everywhere. So it's complicated. So what we did is Etienne did this work and tried to understand what is the signaling. I have to go fast. What he found is that MTCB1 within the mitochondrion is acting through SAC, soluble adenylyl cyclase, which is then activating PKA, and then decreasing the phosphorylation of specific subunits of complex 1. And we used this, yes, so to make a long story short, we wanted to know if we alter this signaling pathway, for example, by altering PKA within the mitochondrion. So we did a mitochondria-specific, MLS means mitochondrial leading sequence, PKA constitutively active. So we inhibit the inhibition, that is, we compensate the inhibition caused by CB1, and we check this in memory. We injected this in the hippocampus, and we use this object recognition memory in which animals have to recognize that one object has changed from one day to the other. And when you have a positive bar, means that you have good memory, and you see that WIN, a CB1 agonist, is fully blocking this memory in control animals. However, if you inject this virus in the hippocampus, the WIN has no effect whatsoever. So if you block the intramitochondrial signaling, you block the effect of cannabinoids in memory. However, this is kind of indirect. So what we wanted to know is, what we wanted to try was really to exclude CB1 from mitochondria. So what we did was that there are programs on internet, MitoProt and PSort, that can tell you what are the theoretical chances of a sequence of a protein to go to mitochondria, to be localized at mitochondria. I don't know how the algorithm works, but this is giving you a theoretical possibility. And CB1 is actually 40%. That is very high for G protein coupled receptor. The other GPCRs are maximum going to 5%, besides some specific, which is also interesting. However, but if you remove the first 22 amino acids in silico on the computer, I remember I was on my bed at home and I was chopping the receptor to see what happens, this is what happens. So the idea is that if you have cells that express CB1, you have both MTCB1 and plasma membrane CB1. However, if you remove the first 22 amino acids, so what we call delta N22, what happens is that you abolish the chances to go to mitochondria. We verified this in reality. And just to summarize, to not lose time, delta N22 CB1 is excluded from mitochondria, is functional, but does not alter mitochondrial functions. So this is a tool really to study the functions of mitochondrial CB1. And what happens is that if we do a viral expression of CB1 and delta N22 CB1 in the hippocampus of CB1 knockout mice, so basically we have a knockout and then we inject either CB1 or delta N22 into the hippocampus. So we make a rescue approach. this is what happens. You take a wild-type mouse, you treat it with Win, you block the memory. This is again the object recognition memory I showed you before. So this is normal control. Then you take the knockout mice, full knockout control conditions, the animals learn, but Win has no effect, right? If you inject the wild-type CB1 in the CB1 knockout mice, you get back the effect of Win. So you rescue the effect of Win. However, if you inject the delta N22 CB1, you do not rescue the effect of Win. So this is telling us that hippocampal MTCB1 is necessary for cannabinoid-induced damnation. So in conclusion, together with the other things that I said before, it's not emerged from cellular and subcellular location, and then we can say that mitochondrial CB1 are present in the brain and mediate bioenergetic and amnesic effects of cannabinoids via different mechanisms. This might be very important from a therapeutic point of view, because you know that there is a huge debate now about cannabis, therapeutic cannabis, and so on. And of course, there are very important side effects that are that are associated with cannabis treatment. So far, I cannot be sure 100%, but so far what we observe is that normally the bad effects seem to involve mitochondrial functions. We have a story with Juan about social interaction that is involving mitochondrial CB1 in astrocytes. We know that catalepsy, loss of ability to move, induced cannabinoids, is probably mediated by mitochondrial CB1. Instead, a positive effect of cannabis, like analgesia, is normal if you don't have CB1 in mitochondria. So it looks that it could be, theoretically, maybe this is interesting for this audience, a way to discriminate between good and bad effects of cannabis. Okay, finally, I would like to thank the lab, and I would like to thank you, all you, for your attention, and I'm sorry if I was a little bit too long.
Thank you very much, Giovanni, very nice talk. We have time maybe for one question, or maybe two, depending on the length. Anybody wants to make any question? Nobody? I think that there is time. I think the results are really, really interesting. So I hope that I'm sure that they have many questions in their minds, but they don't want to make them. I think that it's very, really nice. I was impressed about it's a very short comment or question regarding your first part on the food intake. Do you know whether the hypothalamus contains CB1 receptor? I mean, yes. the neurons yes. from the hypothalamus?
Yes.
Yeah, and are they controlling?
They control. Actually, I mean, CB1 and food intake, it looks like that it is a chain of, yes. In every step of a CB1, of a food intake chain that is making yes. you activate to look for food and to eat. There is a chain of events that is starting from the periphery to the brain and back to the periphery. And CB1 is at many, many steps of this chain. And wherever you block everything.
Yes, I see.
So, yes, hypothalamus is important. This specific GABAergic and glutamatergic thing do not seem to involve the hypothalamus directly.
Oh, okay.
The glutamatergic seems more olfactive. It is control of olfaction, and this is making what we were saying yesterday evening, that one of the reasons why we eat is that our nose tells us to eat.
Okay.
Okay?
Oh, yeah.
Even if we don't know.
Yeah. Very nice. Isabel, if you want to know any question, can we have the microphone, please? Don't worry, we still have 2 minutes. Because he has to leave.
Yeah,
it's okay, this time, don't worry.
I was wondering whether the enzymes required for the production of the endocannabinoids are also present in the mitochondrial membranes.
Yes, yes. We know that mitochondria produce, I mean, at least have endocannabinoids. We know that they degrade because we have one experiment in which we put an inhibitor of degradation, and we saw a very nice in mitochondria isolated, and we saw a very nice increase of endocannabinoids that is perfectly correlating with a decrease of respiration. So more when mitochondria itself have endocannabinoids, less it is respirating. FAH, for example, is one enzyme that is degrading endocannabinoids, is known to be mitochondrial. We know that other enzymes are present in mitochondria and they might be also somewhere else. Yeah, what I think is that, yes, my feeling is that mitochondria produce and use their own endocannabinoids. It's not necessarily something that is coming from somewhere else.
Thank you very much, Gio. Thank you very much also to Isabel. Thank you very much to the audience for staying here, being so patient. I think that I would like to thank you both for coming to Salamanca. Thank you very much. So the symposium is over. Thank you. See you later.